Welcome to a quiet place where we'll explore together one of the most beautiful mysteries of existence, the human brain. Tonight, we're going on a gentle journey deep into the inner world of your mind, where billions of tiny cells work in harmony, creating everything you think, everything you feel, and everything you are. There's no need to rush, no need to memorize. Just listen and let yourself drift as we discover how the soft whisper of electricity becomes the voice of thought. Imagine for a moment that you could shrink down, becoming small enough to wander through the landscape of your own brain. You would find yourself in a vast city, a city unlike any other, where instead of buildings, there are neurons, delicate branching cells that stretch out like ancient trees, reaching toward one another across microscopic spaces. These neurons speak to each other, not with words, but with signals, tiny pulses of electricity that travel along their branches, jumping from cell to cell, creating patterns, creating meaning, creating you. This is not a story of machines or wires or circuits. This is a story of living cells, working together in darkness, creating the light of consciousness, the warmth of emotion, the depth of memory. So settle in, let your breathing slow, and allow yourself to wonder as we begin our journey into the mysterious world of neurons and the electrical impulses that create thought. The living architecture of a neuron. To understand how thought is born, we must first understand the neuron itself, this remarkable cell that forms the foundation of everything your mind can do. A neuron is not a simple cell. It has a shape, a purpose, a kind of elegant architecture that allows it to do something extraordinary, to carry messages across vast distances, at least vast when measured at the scale of a single cell. Picture a neuron as a tree in winter. Its main body, the cell body or soma, sits at the center like a trunk. This is where the nucleus lives, where the essential work of the cell is coordinated, where energy is managed, where life is maintained. From this central body, branches extend outward in all directions. These are the dendrites, delicate, intricate branches that reach out into the space around the neuron, searching, waiting, ready to receive signals from thousands of neighboring cells. The dendrites are listeners. They are covered with tiny receptor sites, like ears turned toward the whispers of other neurons. When chemical messages arrive, these receptors detect them, and the neuron begins to stir, begins to respond. But there is another branch, one that is different from all the others. This is the axon, a single long extension that reaches out from the cell body like a road leading away from a town. The axon is the neuron's voice, its way of sending messages to distant places. Some axons are short, stretching only a tiny fraction of a millimeter. Others are remarkably long. In your body, some axons stretch from your spinal cord all the way down to your toes, a journey of 3 feet or more, all within a single cell. The axon is wrapped in a special coating, a substance called myelin, which looks under a microscope like beads on a string. This myelin sheath acts as insulation, allowing the electrical signal to travel faster, to jump from gap to gap. racing along at speeds that would take your breath away if you could perceive them. At the end of the axon, the neuron branches again, splitting into many terminal branches, each one ending in a small swelling called a synaptic terminal, or bouton, a word borrowed from French meaning button. These terminals hover close to the dendrites of other neurons, not quite touching, separated by a gap so small it can barely be measured. This gap is called the synapse, and it is here, in this microscopic space, that one of the most profound acts in nature takes place. The transfer of a signal from one neuron to another. But before we explore that magical moment, we must understand the signal itself, the electrical impulse that travels along the neuron, giving it life, giving it purpose. The quiet voltage of rest. Even when you are completely still, even when you are here listening in silence, your neurons are never truly at rest. Inside each neuron, inside the watery fluid that fills its membrane, there is an invisible tension, a difference in electrical charge between the inside of the cell and the outside world. This difference exists because of ions, tiny charged particles, atoms of sodium, potassium, chloride, and others, floating in the fluid inside and outside the cell. The neuron's membrane, its outer boundary, is not a simple wall. It is a selective barrier with channels and pumps built into its structure. These pumps work constantly, moving sodium ions out of the cell and potassium ions into the cell, using energy to maintain a careful balance. When a neuron is at rest, not sending a signal, the inside of the cell is more negative than the outside, about 70 millivolts more negative. This is called the resting potential, a state of quiet readiness. Imagine a still pond. The surface is calm, but beneath it, currents flow, energy waits. In the same way, a resting neuron holds potential energy, like a drawn bow, ready to release. This resting voltage is maintained by the sodium-potassium pump, a tiny molecular machine embedded in the cell membrane. It moves 3 sodium ions out. For every 2 potassium ions it brings in, creating an imbalance, a charge difference, a stored voltage. The neuron sits in this state, waiting, listening to the chemical messages arriving at its dendrites. Each message, each neurotransmitter molecule that binds to a receptor, causes a small change in voltage. Some messages are excitatory. They make the inside of the cell slightly less negative, moving it closer to a critical threshold. Other messages are inhibitory. They push the voltage in the opposite direction, making the cell more negative, more resistant to firing. The neuron integrates all these signals, like a quiet accountant, adding up the excitatory inputs, subtracting the inhibitory ones. The dendrites collect these whispers, these tiny voltage changes, and they flow toward the cell body, converging, summing together. And then, if the total reaches a critical point, if the voltage crosses the threshold, something remarkable happens. The action potential, a wave of change. When the voltage inside a neuron reaches approximately negative 55 millivolts, a cascade begins, sudden, irreversible, beautiful in its precision. This is the action potential, the fundamental unit of neural communication, the spark that travels down the axon, carrying information across the landscape of the brain. At the moment the threshold is crossed, voltage-gated sodium channels in the membrane spring open. These channels are sensitive to voltage. When the charge inside the cell becomes less negative, they change shape, opening like gates in a wall. Sodium ions rush in, flowing down their concentration gradient, entering the cell in a sudden flood. This influx of positive charge makes the inside of the cell rapidly become positive in less than a millisecond. The voltage swings from negative 70 millivolts to positive 30 millivolts. This is depolarization, a moment when the careful balance is overthrown, when the neuron's interior becomes electrically positive. But the change doesn't stop there. Almost immediately, the sodium channels close and potassium channels open. Potassium ions flow out of the cell, carrying positive charge away, restoring the negative interior. This is repolarization, the return to balance. Within another millisecond, the voltage drops back below the resting potential, briefly overshooting into hyperpolarization before the sodium-potassium pumps restore the original state. The entire event, from threshold to return, takes only 2 or 3 milliseconds. A brief flicker, a momentary reversal of charge, and then equilibrium restored. But here is the beauty of it. This change in voltage at one point on the axon triggers the same change in the adjacent section. The opening of sodium channels in one spot causes the voltage nearby to reach threshold, which opens more sodium channels, which causes depolarization there, which spreads to the next section. And so the action potential propagates, like a wave traveling along a rope, like ripples spreading across water. The wave moves down the axon, away from the cell body, toward the terminals, carrying its message forward. In unmyelinated axons, this wave flows smoothly and continuously, traveling at speeds of about 1 meter per second. But in myelinated axons, those wrapped in their insulating sheath, something different occurs. The myelin prevents ions from crossing the membrane. The action potential cannot occur beneath the myelin. Instead, it jumps from one gap in the sheath to the next. These gaps are called nodes of Ranvier, and the jumping process is called saltatory conduction. This jumping allows the signal to travel much faster, up to 100 meters per second, faster than a race car on a highway, all within the microscopic corridors of your nervous system. The action potential is all or nothing. It either happens completely or it doesn't happen at all. There is no partial action potential, no weak or strong version. Once threshold is reached, the cascade proceeds with fixed intensity. This might seem limiting, but it's actually elegant. The strength of the signal is not in its size, but in its frequency. A neuron experiencing strong stimulation fires action potentials more rapidly, creating a pattern, a rhythm, a code. The synaptic conversation. Now the action potential has traveled the length of the axon, racing along its insulated path, jumping from node to node. It arrives at the axon terminals, those small button-like endings that hover near the dendrites of other neurons. Here, in this tiny space, something profound is about to occur. The action potential cannot jump directly from one neuron to the next. There is a gap, the synaptic cleft, a space of about 20 nanometers, impossibly small, yet enough to prevent electrical current from flowing across. So the signal must change form, from electricity to chemistry, and then back to electricity again. When the action potential reaches the synaptic terminal, it causes voltage-gated calcium channels to open. Calcium ions flow into the terminal, and their presence triggers a cascade within the cell. Inside the terminal, there are small spheres called synaptic vesicles. Each one is like a tiny bubble filled with molecules of neurotransmitter, thousands of molecules per vesicle, waiting to be released. The influx of calcium causes these vesicles to move toward the membrane, to fuse with it, to open, releasing their contents into the synaptic cleft. This process is called exocytosis, and it happens with remarkable speed. Within a fraction of a millisecond, neurotransmitter molecules spill into the gap, diffusing rapidly across the space. The synaptic cleft, though tiny, is now filled with these chemical messengers. Each molecule drifts, carried by random thermal motion, bouncing off water molecules until it encounters a receptor on the other side, on the membrane of the receiving neuron's dendrite. Imagine standing on one shore of a narrow river and tossing a message in a bottle to someone on the other side. The bottle floats across, is caught, and the message is read. This is the essence of synaptic transmission. The neurotransmitter binds to its receptor. Each type of neurotransmitter has specific receptors, fitting together like a key in a lock. When they bind, the receptor changes shape. Some receptors are ionotropic, meaning they are ion channels themselves. When the neurotransmitter binds, the channel opens, allowing ions to flow, changing the voltage in the receiving neuron. If sodium or calcium ions flow in, the receiving neuron becomes less negative. It moves toward threshold. This is an excitatory postsynaptic potential, a small push toward firing. If chloride ions flow in or potassium flows out, the cell becomes more negative, moving away from threshold. This is an inhibitory postsynaptic potential, a gentle restraint. Other receptors are metabotropic. They don't form channels themselves. Instead, when the neurotransmitter binds, the receptor activates proteins inside the cell. These proteins trigger cascades of chemical reactions, modulating the cell's behavior over longer timescales, changing its sensitivity, its metabolism, its very nature. The beauty of this system is its flexibility. A single neuron receives inputs from thousands of other neurons, some excitatory, some inhibitory. Each synapse is a vote. Each neurotransmitter release is a whisper of influence. And all these whispers are integrated, summed together in the receiving neuron's cell body. If the total excitation exceeds the total inhibition by enough, if threshold is reached, then this neuron fires its own action potential, continuing the cascade, passing the signal onward to the next cells in the network. The chemistry of connection. There are many neurotransmitters in the brain, each with its own character, its own role in the symphony of thought. Glutamate is the most common excitatory neurotransmitter. It is the language of learning, of activation. When glutamate is released, it tells the receiving neuron to become more active, to pay attention, to strengthen the connection. GABA, gamma-aminobutyric acid, is the primary inhibitory neurotransmitter. It calms, it quiets. When GABA binds to its receptors, it helps prevent overstimulation. It maintains balance. Without GABA, the brain would cascade into chaos, into seizure. Then there are the modulatory neurotransmitters, chemicals that don't just excite or inhibit, but change the way neurons respond. Dopamine, released by neurons in the midbrain, is associated with reward, with motivation, with the sense that something good is happening or about to happen. It modulates the strength of synaptic connections. It teaches the brain what to seek, what to remember. Serotonin, spreading from the RAF nuclei, influences mood, sleep, appetite. It creates a sense of well-being when present in the right amounts. Its absence is linked to depression, to anxiety, to restlessness. Norepinephrine, also called noradrenaline, enhances alertness, attention. It prepares the brain for action, for response. Its release says, "This matters. Pay attention." Acetylcholine, one of the first neurotransmitters to be discovered, is involved in learning, in memory formation, in the activation of muscles. It bridges thought and action. Each of these chemicals, and dozens of others, adds a different color to the palette of neural communication. Together, they create the complex, layered experience of consciousness, and the neurotransmitters don't simply act and disappear. After they bind to their receptors, they must be cleared from the synaptic cleft, removed so the signal can end, so the synapse can reset and prepare for the next message. Some neurotransmitters are taken back into the releasing neuron through transporter proteins, a process called reuptake. Others are broken down by enzymes in the synaptic cleft, dissolved into harmless fragments that are recycled. This cleanup is essential. Without it, neurotransmitters would accumulate, signals would blur together, communication would fail. Many psychiatric medications work by affecting these cleanup processes, by prolonging the presence of neurotransmitters in the synapse, allowing their effects to last longer. Networks of light. A single neuron, with its dendrites receiving inputs and its axon sending outputs, is remarkable. But a single neuron does not create thought. Thought emerges from networks, from populations of neurons connected in patterns, from the collective behavior of millions or billions of cells acting in concert. Consider that a typical neuron in the cerebral cortex receives inputs from about 10,000 other neurons and sends outputs to another 10,000. This creates a web of connectivity so dense, so intricate, that it defies easy description. Imagine a city at night, seen from above. Every light is a neuron. Every connection between lights is a synapse. When neurons fire, they light up, and the pattern of illumination creates shapes. Waves, clusters of activity that ripple through the network. This is not random. The connections between neurons are not arbitrary. They form circuits, pathways that have been shaped by experience, by development, by the unfolding of genetic instructions. Some circuits are simple. Sensory neurons in your skin connect to neurons in your spinal cord, which connect to motor neurons, which cause muscles to contract. This is a reflex arc. A tap on your knee, and your leg kicks before you even consciously register the sensation. Other circuits are vastly more complex, involving loops of connections, feedback, inhibition, modulation. The visual cortex, for example, contains dozens of interconnected regions, each processing different aspects of the visual scene: edges, colors, motion, faces. Each region sends information to others, receives information back, creating a dynamic, ever-shifting pattern of activity. What's remarkable is that these patterns are information. When you see a face, certain neurons fire in a specific pattern. When you recall that face later, a similar pattern reactivates, though perhaps not exactly the same. The memory is the pattern. The recognition is the pattern. The face itself, as it exists in your mind, is nothing more and nothing less than this coordinated firing of neurons. This is called neural coding. The idea that information in the brain is represented by patterns of neural activity. It's not that a single neuron represents a single concept. It's that populations of neurons, firing together in specific rhythms and relationships, create meaning. Some patterns are sparse, involving just a few highly specific neurons firing. These might represent precise, detailed information, like the memory of a particular moment, a particular sensation. Other patterns are distributed, involving broad regions of the cortex, millions of neurons contributing their small voices to a collective state. These might represent more abstract concepts, emotions, contexts, the general sense of a situation. And these patterns interact. Patterns in one region influence patterns in another. Activity in the visual cortex affects activity in memory regions, which affects activity in emotional centers, which feeds back to sensory areas, changing what you notice, what you attend to. The brain is not a passive receiver of information. It is an active predictor, constantly generating expectations about what will happen next, comparing these predictions to incoming sensory data, adjusting its models when surprised. This predictive processing occurs through networks that extend from higher cognitive regions down to primary sensory areas. Top-down signals carry predictions. Bottom-up signals carry prediction errors. The meeting of these signals, the negotiation between expectation and reality, this is perception. Synaptic plasticity, the sculpting of thought. The networks of the brain are not fixed. They change constantly, with every experience, with every thought. This capacity for change is called plasticity, and it is perhaps the most important property of the nervous system. When two neurons fire together repeatedly, when the presynaptic neuron's action potentials consistently lead to activity in the postsynaptic neuron, the synapse between them strengthens. This is Hebb's rule, often summarized as neurons that fire together, wire together. When a synaptic connection is used frequently, it becomes more efficient. The presynaptic terminal releases more neurotransmitter. The postsynaptic membrane expresses more receptors. The connection becomes faster, more reliable. This strengthening is called long-term potentiation, or LTP, and it can last for hours, days, or even a lifetime. The opposite also occurs. When a synapse is rarely used, when the presynaptic neuron fires but the postsynaptic neuron remains quiet, the connection weakens. This is long-term depression, or LTD. The synapse becomes less effective. Its influence diminishes. Through this continuous process of strengthening and weakening, the brain sculpts its circuits, reinforcing pathways that are useful, allowing unused pathways to fade. This is how learning happens. When you practice a skill, certain neural pathways are activated repeatedly. The synapses along those pathways strengthen, The action becomes easier, more automatic. What once required conscious effort becomes fluid, natural. When you form a memory, patterns of synaptic connections are strengthened in regions of the hippocampus and cortex. The memory is encoded as a change in the physical structure of the brain. A network that didn't exist before now exists. A pattern that couldn't be activated before can now be called up at will. Plasticity occurs at multiple timescales. Some changes happen in seconds or minutes, like the working memory that holds information while you complete a task. Other changes take days or weeks, like the consolidation of memories from short-term to long-term storage. Still others unfold over months and years, like the rewiring that occurs during recovery from brain injury. Even in adulthood, even in old age, the brain retains remarkable plasticity. New synapses can form, new dendrites can grow. The networks can reorganize, adapting to new demands, new environments, new challenges. This means that who you are is not fixed. Your patterns of thought, your habits, your emotional responses, all of these exist as networks of synaptic connections, and these networks can change. Every thought you think, every action you take, subtly alters the physical structure of your brain. You are, in a very real sense, constantly sculpting yourself, creating yourself anew with each passing moment. From neurons to networks to thought, we've journeyed from the single neuron, with its dendrites and axon, its resting potential and action potential, to the synapse, where chemistry bridges the gap between cells, to networks, where millions of neurons create patterns that represent information. But how do these patterns become thought? This is one of the deepest questions in neuroscience, and we don't have a complete answer. But we have glimpses, pieces of understanding that hint at the whole. Thought seems to arise from the integration of activity across many brain regions. When you think about a familiar face, visual regions activate. Remembering the appearance, auditory regions might activate if you recall their voice. Emotional regions respond with feelings associated with that person. Language regions might bring up their name. All of these separate streams of activity, occurring in different parts of the brain, somehow cohere into a single unified experience. This integration likely involves oscillations, rhythmic patterns of neural firing that synchronize activity across distant regions. Neurons in different areas fire in phase with each other, creating temporal patterns that bind their activity together. When you're awake and alert, your brain produces fast oscillations, gamma waves cycling at 30 to 100 times per second. These rapid rhythms might serve as a kind of temporal glue, allowing information from different regions to be combined, to be compared, to be synthesized into coherent thoughts. Slower oscillations play different roles. Alpha waves, around 10 cycles per second, are prominent when you're relaxed with eyes closed. They might serve to inhibit irrelevant sensory information, to quiet the noise. Theta waves, around 4 to 8 cycles per second, are important for memory formation. They appear in the hippocampus during learning, coordinating the transfer of information into long-term storage. Delta waves, the slowest, less than 4 cycles per second, dominate during deep sleep, when the brain consolidates memories, repairs itself. prepares for the next day. These oscillations don't occur in isolation. They nest within each other. Faster rhythms ride on top of slower rhythms, creating complex temporal structures. These structures might be the substrate of consciousness itself, the rhythmic dance of neural activity that creates the flow of subjective experience. Attention seems to work by enhancing certain patterns while suppressing others. When you focus on something, neurons representing that thing fire more strongly, more synchronously. Meanwhile, neurons representing irrelevant information are inhibited. Their activity is dampened. This selective enhancement and suppression creates a foreground and background in neural space. What you attend to becomes bright, vivid, detailed. What you ignore fades into the periphery, barely registered, and this process is dynamic. Your attention can shift, redirected by unexpected events, by goals, by interests. As it shifts, the patterns of activity shift. Different neurons take the foreground. Different patterns dominate the network. Consciousness itself, that most mysterious of phenomena, appears to involve specific patterns of connectivity and activity. Certain networks in the brain, particularly those connecting the thalamus, cortex, and association areas, seem to be essential for conscious awareness. When these networks are disrupted by anesthesia, by sleep, by injury, consciousness fades or vanishes. The neural activity doesn't stop, but it becomes disconnected, uncoordinated. The integration that creates unified experience is lost. There's a theory called integrated information theory, which suggests that consciousness arises when a system has both high complexity and high integration, when it contains many differentiated parts that are also unified into a whole. The brain, with its billions of neurons connected in intricate patterns, has exactly this property. Another theory, the global workspace theory, suggests that consciousness is like a stage where information from various specialized processors can be broadcast to the rest of the system. When a neural pattern enters this global workspace, it becomes conscious, available for reasoning, planning, verbal report. These theories are different, but they share a common insight: that consciousness is not located in any single place. It is not a thing, but a process, an emergent property of complex, integrated neural dynamics. The resting brain, a symphony in the dark. Even when you are at rest, when you are not engaged in any particular task, your brain is profoundly active. This resting state activity is not noise. It is structured, organized, meaningful. There are networks of regions that activate together even when you're doing nothing in particular. These are called resting state networks, or intrinsic connectivity networks. The most prominent is the default mode network, a set of regions including the medial prefrontal cortex, posterior cingulate cortex, and parts of the parietal lobes. This network activates when you're daydreaming, mind-wandering, thinking about yourself and others. It seems to be involved in self-referential thought, in imagining, in recalling personal memories. When you engage in a focused task, the default mode network quiets, and other networks activate: the dorsal attention network for focused external attention, the executive control network for planning and decision-making, But the default mode network never completely shuts off. It continues its quiet background activity, and researchers believe this activity is important. It might be involved in consolidating memories, in making connections between ideas, in the kind of creative thinking that occurs when you're not trying. The brain at rest consumes almost as much energy as the brain actively engaged in a task. About 20% of the body's total energy budget. This enormous expenditure in apparent idleness suggests that this spontaneous activity is crucial, that the brain is always working, always maintaining its networks, always preparing for the next moment. There's also evidence that sleep, particularly deep, slow-wave sleep and REM sleep, is when the brain performs essential maintenance. Synapses that were strengthened during the day are selectively weakened if they're not important. This is called synaptic downscaling, and it prevents the brain from becoming saturated, from losing the ability to form new memories. During sleep, the brain also clears out metabolic waste products that accumulate during waking. The glymphatic system, a network of channels around blood vessels, flushes fluid through the brain tissue, carrying away toxins. This cleansing process is most active during sleep, when neurons shrink slightly, opening up the spaces between cells. And dreams, those strange, vivid experiences during REM sleep, may serve to consolidate emotional memories, to work through unresolved feelings, to integrate new experiences with existing knowledge. The hippocampus and cortex show patterns of activity during sleep that resemble the patterns from waking experience, as if the brain is replaying the day, cementing important moments into long-term storage. The living whole. We've explored neurons at many scales, from the molecular machinery of ion channels to the electrical cascade of the action potential, to the chemical conversation at the synapse, to the networks of millions of cells, to the global patterns that create thought and consciousness. At each level, there is beauty. There is precision. There is a kind of elegant logic. But the brain is more than the sum of its parts. It is a living system, constantly adapting, constantly changing. It cannot be understood purely by breaking it into components. The neurons themselves are alive. They require energy. They have metabolisms. They are born. They grow. They sometimes die. Throughout your life, your brain generates new neurons, particularly in the hippocampus. This neurogenesis continues even in old age, contributing to the brain's capacity for change. The neurons are supported by other cells, glial cells, once thought to be mere support structures, are now known to be active participants in neural function. Astrocytes regulate the chemical environment around synapses, taking up excess neurotransmitter, supplying neurons with nutrients. Oligodendrocytes create the myelin sheaths. Microglia serve as immune cells, protecting the brain from infection and clearing away cellular debris. All of these cells work together in a community, each one contributing to the health and function of the whole. And the brain is embedded in the body. It receives constant signals from the body's internal state. Hormones from the endocrine system affect neural activity, changing mood, motivation, attention. Signals from the gut, from the immune system, from the cardiovascular system, all influence the brain's state. The brain, in turn, controls the body, regulating heart rate, breathing, digestion, movement. It maintains homeostasis, the stable internal environment necessary for life. This bidirectional communication between brain and body means that mental states affect physical health, and physical states affect mental experience. Chronic stress changes brain structure. Depression affects immune function. Mindfulness practice can reduce inflammation. The barrier between mind and body is not as clear as we once thought. Your thoughts, your emotions, your memories, all exist as patterns of neural activity. But these patterns are instantiated in living tissue, in cells that require oxygen and glucose, that generate heat, that produce waste. The substrate of consciousness is biological, mortal, vulnerable, and yet, within this biological substrate, something extraordinary occurs. The mere physical movements of ions across membranes, the binding of molecules to receptors, the propagation of voltage changes along axons, somehow gives rise to the subjective quality of experience. The redness of red, the sound of music, the feeling of joy, the taste of sweetness. These qualia, these subjective experiences, emerge from objective physical processes, from the dance of electrons and ions, from the collective behavior of billions of cells, comes the inner life of the mind. This is the hard problem of consciousness. The question of how and why physical processes produce subjective experience. And while we can trace the neural correlates, the patterns of activity associated with different experiences, we still don't fully understand the bridge between the physical and the phenomenal. Perhaps consciousness is fundamental, a basic property of certain types of information processing. Or perhaps it emerges naturally from sufficient complexity and integration. Or perhaps there's a deeper principle we haven't yet discovered. For now, we can appreciate the mystery. We can marvel at the fact that the universe, through the evolution of nervous systems, has created matter that is aware of itself. Atoms that were forged in stars, arranged into patterns that can contemplate the stars. The individual and the universal. Your brain is unique. The exact pattern of connections in your brain has never existed before and will never exist again. Every experience you've had, every thought you've thought, has left its mark on your neural networks, has shaped the person you are. And yet, the principles are universal. Every human brain operates by the same basic mechanisms. Neurons fire action potentials. Synapses release neurotransmitters. Networks create patterns. The differences between minds are differences of detail, of connection strength, of network architecture, not differences of fundamental principle. This means that every mind shares a common foundation, that at the deepest level, the experience of being human, of being conscious, of thinking and feeling, is something we all share. When you interact with another person, you are interacting with another network of neurons, shaped by different experiences, but operating by the same rules. Their thoughts are electrical patterns, just like yours. Their feelings are chemical messages, just like yours. This shared basis of neural function is what makes empathy possible, makes communication possible, makes society possible. You can understand another person's experience because their brain creates experience in fundamentally the same way yours does. And this understanding, this recognition of the neural basis of mind, doesn't diminish the human experience. It deepens it. To know that love is patterns of activity in the limbic system, that memory is strengthened synapses in the hippocampus, that decision-making involves oscillations in the prefrontal cortex. This doesn't make these experiences less real, less meaningful, less profound. If anything, it makes them more wondrous. That such complexity, such depth, such richness of experience can arise from the coordinated activity of biological cells. This is not reductionist. This is miraculous. Your capacity for language, for abstract thought, for moral reasoning, for art, for science, all of this emerges from neural networks that were shaped by millions of years of evolution, refined by thousands of generations, and sculpted by your unique lifetime of experience. You are the latest expression of a process that began billions of years ago, when the first simple nervous systems evolved in ancient organisms. Systems that could detect light, respond to touch, coordinate movement. From those humble beginnings, through endless iterations of variation and selection, emerged the human brain, with its 100 billion neurons and its capacity for self-reflection. And the process isn't finished. Your brain continues to change, continues to grow new connections, to sculpt its networks in response to experience. You are not a fixed entity. You are a process, an ongoing pattern of neural activity that persists through time, that changes while remaining somehow the same. Closing, rest in the mystery. We've traveled far together tonight, from the resting potential of a single neuron to the vast networks that create consciousness. We've seen how electricity becomes chemistry, how chemistry becomes electricity again, how patterns of activity become patterns of thought. We've glimpsed the machinery of the mind, the ion channels and action potentials, the neurotransmitters and synapses, the oscillations and networks. But more than machinery, we've witnessed a kind of beauty, an elegance in the way these systems work together. Your brain, humming quietly in darkness behind your eyes, is performing these processes right now. Neurons are firing, synapses are transmitting, networks are integrating information, thoughts are arising, dissolving, being replaced by new thoughts. And all of this intricate activity is happening effortlessly, without your conscious direction. The neurons know what to do. The patterns form themselves. The system regulates itself, maintains itself, heals itself. There's something comforting in this, in knowing that beneath the level of conscious awareness, your brain is taking care of you, managing the countless processes necessary for life, for thought, for experience. As you rest now, as you drift towards sleep, your brain will continue its work, consolidating memories from today, strengthening important connections, weakening others, preparing for tomorrow. The slow oscillations of deep sleep will wash through your cortex like gentle waves. The glymphatic system will clear away the metabolic debris of the day. The neurons will rest, recover, reset, and in the morning, when you wake, the networks will be refreshed, ready to process new information, to form new thoughts, to create new patterns of experience. This is the gift of the brain, this biological miracle that creates your inner world, that allows you to perceive, to think, to feel, to be. Rest now, in the knowledge that you are a living process, a pattern of electrical and chemical activity that has become aware of itself. You are the universe experiencing itself. Neurons contemplating neurons. Consciousness reflecting on consciousness. Let the mystery of it wash over you. Let yourself feel the wonder of existence, and as you settle into rest, know that your brain will continue its ancient work, pulsing with gentle electricity, whispering with chemical messengers, weaving the fabric of your mind. Sleep well and let the neurons do their dance in the darkness, creating dreams, consolidating memories, preparing you for another day of thought, another day of being alive, another day of being you. The journey of the neuron continues in the quiet space within your skull, where electricity becomes thought, where chemistry becomes consciousness, where the mysterious machinery of the mind creates the experience of being human. Rest now and trust the process. The neurons know the way.